Hello and welcome to my channel. So today we are going to discuss cancer, some of its mechanisms and ways to prevent it. Since we know that cancer is abnormal growth of cell, so before going further, let's discuss how normally cells grow. So normally these cells they divide to produce new cells, and they should only do this when needed, such as for growth and to repair and renew tissue. Now, normal cells divide via cell cycle. They divide at an appropriate rate for the area of the body. Some have a high rate of cell turnover, for example, skin, liver, bone marrow, and tissues lining the digestive system, lungs, bladder, and all. And so, what happens now? Normal cells they are under the control of an internal clock that determines when the cell should rest, when it should divide, and when it should die. So, they are under a control. Normal cells also have housekeeping mechanisms that should stop abnormal cell behavior, and if they can't correct an abnormality, because we have a lot of tumor suppressor genes present inside, which we will be discussing further, they should tell the cell to self destruct. So the problem with cancer is they disobey these rules of cell normality, and it's a large group of disease that can start in almost any organ or tissue of the body. That is the problem with cancer. And we have a group of tumor suppressor genes present inside. For example, we will discuss p53, p21 and all, which help regulate the cell cycle. So it is a disease in which cells change and grow uncontrollably. There are more than 100 different type of cancers, and the most common type of cancer, they are colorectal, stomach, intestine, lung, prostate in men, breast cancer, thyroid cancer, and cervical cancer in women, and the list goes on. So let's see how a normal cell differs from a cancer cell. So if you see here this graphic, this representation of a normal cell, we can see that the normal cells they are growing within limits, within the boundaries, and they are attached to the substratum. And so this property is called contact inhibition, which is not obeyed by the cancer cells, because cancer cells they are growing abruptly, and they don't have any, they don't take care of any boundaries. Normal cells they follow contact inhibition. That means if they start touching each other, they don't grow further. Whereas the cancer cells they don't follow this property of contact inhibition. Irrespective that whether they are touching each other, they keep on growing and keep on growing. So I have a previous video, I'll put that in my description box, where I have discussed differences between and properties of cancer cells. So now these cancer cells, this property is called metastasis of cancer, where uh, they don't remain localized, they cross the boundaries, they go to the bloodstream, and they travel through the bloodstream and they reach different locations, and they disrupt the boundary and the linings, and that's how they spread. Now let's discuss causes of cancer. So the most common cause of cancer is mutation, genetic mutations. 5% are acquired, so they are hereditary mutations, and 95% uh, they are acquired mutations from life habits and environmental causes. So 5% we get from our parents, grandparents, and 95% are through our lifestyle. So let me tell you one thing here. This is called predisposed genes. When you already are on a risk, right? That means if your parents, your grandparents, or someone on in your family tree has cancer, so that means you are on 5% risk and your genes are predisposed. And rest 95% you acquire from your lifestyle. That can be anything. If you're a chain smoker, or if you have a stressful job, that can be anything. So acquired mutations from lifestyle habits and environmental causes can be due to smoking, alcohol, bad habits, radiation and pollution, certain chemicals and compounds, or if you are exposed to certain viruses like HIV, HPV, HTLV, that degrades the immunity. So low chronic immunity is one of the cause. So these are the causes through which we can acquire cancer. So human body has over 10 trillion cells, and which keep on dividing, Our cells, they are born, they live, and they die. We have a strict control, as I mentioned earlier, which are called housekeeping genes, tumor suppressor genes we have inside our body. That is a strict control which makes sure that anything which is growing abruptly, it should be stopped. So these genes, p53 gene, they play an important part on cells life control. And the protein which is expressed by these genes are called p53 protein. It is responsible for keeping a check if cells are going uh, beyond the limit and they are dividing abruptly. So it induces apoptosis. Apoptosis means natural cell death, programmed cell death. Now, origin of cancer is mainly due to DNA alterations. Uh, DNA alterations are the main causes of cancer. So what happens now? One of the reason is when you have a defective p53 gene, which caused expressed defective p53 protein. Since we have a defective p53 protein, it cannot signal apoptosis process here, and hence malignant cells, they stay immortal. And as I mentioned, they don't follow that property of contact inhibition and they keep on growing. That's why they are immortal. They disobey the rule of contact inhibition. They start reproducing uncontrollably. Thus, they invade and destroy nearby tissues and organs. So let's see the stages of cancer, how it spreads. Since from a normal cell, we have three categories, like a normal cell, tumor, and then a cancer. So what is a primary tumorigenesis? Let's see first. So the transformed cells which are not normal anymore, they release abnormal cytokines. Like these cytokines are low molecular weight communicating molecules, signaling molecules which help in communicating between immunological cells. So they attract macrophages and other immunological cells in the body, because macrophages, they invade, or they eat up the foreign pathogens. They try to destruct the infected cells, anything which is affecting our body. So they phagocyte the transformed cells, and the immune system keeps reacting, but it is ineffective, obviously, because of the tumor growth, because the tumor growth right now is massive. So tumor cells, they release proteins which are known as tumor markers on the bloodstream. And most of the primary tumors, they can be detected by tumor markers and image diagnosis, and that's how it can be detected at an early stage uh, uh, by this. Next stage is metastasis. That is when cancer, when it starts spreading, it's crossing the boundaries, it's entering the bloodstream. So these macrophages, they release certain metalloproteins. These are called metalloproteins because they bind to metal ions. So MMP1 and MMP2, which binds to zinc ions, they are phagocyting tumor cells. They release MMP1 and MMP2. So zinc reacts with them and ends up crossing the bloodstream. These are the certain drugs which are used against tumors. So there are three ways through which we can neutralize tumor cells: blocking receptors, blocking signaling compounds, and blocking the DNA. So before moving further, let's see how cell signaling in normal cells work. So let's assume that this is the cell surface, and these are the receptors on the cell surface. Receptors are specific for the ligand. So this is ligand, this is receptor. After receptor ligand binding, the signal goes inside, and the second step is transduction of signaling compounds. So when signaling compounds transduction, that means signal goes from one X to Y, Y to Z, and so on. And finally, the response happens. So if the blockage takes place at the first step itself, that is receptor blocking. So receptor blocking is done by specific monoclonal antibodies, and it inhibits the action of tumor signaling compounds, killing only the tumor cell. That is the second step, blocks it. The blocking of signaling compounds or DNA is done by nanoparticles, which are composed of specific chemotherapy, docking the receptor, and these ways. And finally, through these ways, these ways can act as drugs. We can work on some drugs which can act against tumors, because uh, biodegradable polymers, these are also options. And nanoparticles, they carry drugs with specific targets, signaling compounds or DNA, and hence they kill specifically the tumor cells. Last is to check, you know, to make sure that we know the best prevention methods against cancer. And the best method is to early detect it. So we should be aware of symptoms like lump or swelling anywhere in the body, unusual bleeding, weight loss that is not related to dieting, long lasting pain, cough, change in voice or breathlessness, or cough which doesn't last for more than 3 weeks, which is more than 3 weeks, sore or spot that doesn't heal, or change in bowel movements. So ultimately, with basic care and adequate treatment, cancer can be treated. Besides, a change in diet can also help prevent cancer. There are a lot of supporting research papers which support mechanisms that repair DNA damage. For example, a diet high in vegetables and fruits can provide antioxidants that help protect DNA. So we should encourage people to have balanced diet, encourage them to have at least one portion of leafy green vegetables like broccoli, because they have a lot of antioxidant properties within them. Also, they should be encouraged to have rainbow diets with colorful vegetables, a lot of pigments within them, like carotenoids, red, purple, and other pigments. These are very good antioxidants. So yellow and red bell peppers and all. And besides, diet also helps prevent because it influences hormone balance. So dietary patterns can influence where is hormones and can balance it, which can also help in reducing the DNA alterations, and it can influence blood sugar imbalances also. It can keep a check on that. So people with diabetes are more likely to develop certain cancers, and a reoccurrence is also more likely. So a healthy lifestyle, a healthy diet can help keep a check on such symptoms. So that's all for the video today, and I hope you liked my video. Do subscribe to my channel, and thanks.